The movement of water across the cell membrane will result in a volume change in the cell, that is the intracellular fluid, or a volume change in the extracellular fluid. And the reason for this is that even with diffusion operating to move water and electrolytes, the cell also uses active transport to carefully maintain the concentration of its internal environment and the electrolytes that are in that internal environment. Water will then follow by osmosis the concentration changes of those electrolytes. The volume changes that occur during this movement of water is referred to as tonicity, and most of the time cells are in an isotonic environment. The concentration of water and electrolytes are equal inside and outside of the cell, and therefore no net movement of water will occur, meaning that water moves into the cell at an equal rate that it moves out of the cell. So it just happens to move in and out randomly. Cells in a hypertonic environment will lose water to their surroundings because electrolyte or solute concentrations are higher on the outside of the cell than on the inside, and water will then exit the cell by osmosis. Cells in a hypotonic environment will gain water from their surroundings because solute concentrations are higher on the inside of the cell than they are on the outside, and water will then enter the cell by osmosis. So figure 3.8 shows us the volume changes of cells when exposed to hypertonic, isotonic, and hypotonic environments. In a hypertonic environment, the electrolyte or solute concentrations outside the cell are greater than on the inside of the cell, and water will then exit the cell by osmosis, causing the cells to shrink. In an isotonic environment, solute concentrations are equal inside the cell and outside the cell, and therefore by extension so are the water concentrations. Therefore water will exit the cell at the same rate that it enters the cell. In a hypotonic environment, the solute concentrations outside the cell are lower than on the inside, and water will enter the cell by osmosis. And we see that if too much water enters the cell could potentially burst. This figure illustrates the solute concentrations in the different tonicities. For the hypertonic solution, the solute concentrations outside the cell are greater than on the inside, and therefore we see water exiting the cell. For hypotonic solutions in the center, the solute concentrations outside the cell are lower than on the inside, and in that diagram we see water entering the cell. For isotonic solutions, the solute concentrations inside and outside the cell are equal, and therefore water equally moves into and out of the cell. The term iso in isotonic means same or same tonicity.